So this kicks off actually our first research seminar of this semester for the Friday Ecology Seminar Series. Ben is on sabbatical, so Dan, Ruman, and I are going to be serving as hosts. Today we have Eric Duell. He is a new postdoc here, like really literally started a few weeks ago. So it's a great opportunity for him to meet us and see many of us and for us to learn about his expertise. Peggy is going to introduce Eric, but before that, Helen has an announcement.
Thank you very much. So I just wanted to mention for the EEB grad students that are on, I had sent an email about possibilities for summer field courses. This is something we've had erratically in the past, but we'd like to have more regularly of them and also support grad students. We got kind of a late start on this, but I've had recent conversations with Mark Mort that suggests that financial support could be there. So if you're interested, please contact me soon. I think I had said by the 13th. And I'm glad to chat about options. One couple little tiny things. We had mentioned a 2-credit course in the email, but 1-credit courses could also be an option. And it doesn't have to be all at the field station. For example, if you were an aquatic ecologist and wanted to do things at the river, that's fine too. So let me know if you're interested.
Helen, just to clarify, this is an opportunity to teach a summer course, correct?
Yes, to teach a summer course.
Thank you. So Jim is at a search committee meeting right now, so I have the honor of introducing Eric Duell, a new postdoc in our lab. Eric received his PhD, master's, and undergraduate degrees from Oklahoma State University, where he worked with Dr. Gail Wilson, a leader in mycorrhizal ecology. Eric has built a strong publication record that includes many publications focused on the relationships between plants and associated mycorrhizal fungi in native prairie plant species, with invasive plants, and in sustainable agricultural settings. We're fortunate to have Eric join us at KU. He will be working on an EPSCoR maps project connecting the plant and soil microbiomes to aquatic systems. He has a USDA grant pending that will focus on the impacts of the soil microbiome and plant diversity on restoration of soil health. Today, he will be discussing his research on Asclepias species. His talk is entitled From Soil Symbionts to Disturbances, Eco-evolutionary Insights into Asclepias. Thank you. And thank you, Eric.
All right, I'll go ahead and share my screen here. All right. All right, well, thank you for that introduction, Peggy. And without further delay, I'll go ahead and jump right into it. So just first, a brief overview of what I'll be discussing today. These are two corresponding chapters of my recent dissertation that I finished back in November of 2021. The first part of my talk will be titled Interspecific Variation in Milkweed Responses to Arbuscular Mycorrhizal Fungi, and I'll follow that up with long-term effects of fire, grazing, and drought on milkweed abundances in the tallgrass prairie. So for those of you that aren't familiar with arbuscular mycorrhizal fungi, or AM fungi, as you'll likely hear me call them throughout this talk, I just want to give a brief overview of this relationship. In this mutualism, this typical mutualism, the AM fungi are penetrating the root cortex and extending these hyphal structures out into deep soil pockets and soil pores that the plants can't access themselves through their root system. The fungal hyphae are uptaking nutrients, usually in the form of inorganic phosphorus, and shuttling that back to the plant through the root system. And in return, the plant's allocating sometimes up to a quarter of the carbohydrates it produces via photosynthesis to sustaining the fungi, because the fungi cannot produce carbon on their own. Now, this relationship is incredibly important, and a testament to that is the fact that between 80 and 90% of Earth's terrestrial vascular flora form some degree of relationship or association with arbuscular mycorrhizal fungi. So this relationship. It's ubiquitous. It can be found across the planet in most ice-free terrestrial surfaces. And so this has led to a growing body of research that is set out to identify or to be better able to predict how dependent or how responsive a plant species may or may not be to arbuscular mycorrhizal fungi without having to conduct these greenhouse assays, just because it's unrealistic to do this for every plant species on the planet. And so to date, it's pretty well accepted that functionally similar species have similar dependencies or responses to AM fungi. And the two that stick out in my mind are C3 versus C4 grasses. So grasses that utilize a C4 photosynthetic pathway are typically much more responsive or dependent on this symbiosis compared to C3 counterparts. On top of that, perennial plant species are typically much more dependent or responsive than annual plant species. And this pattern holds true for a number of other either life history or functional traits. And so we know there's often little variation within functional groups, and it's thought that there's even less variation within taxa. So within these more closely related species, we don't expect, or previous research and literature indicates that we shouldn't expect much variation. And I always refer back to my PhD advisor's work at Konza, Prairie in the late 1990s, where she conducted assays on roughly 100 different plant species from across Konza Prairie to test them for their mycorrhizal responsiveness. And although she didn't take any kind of phylogenetic approach, just combing through the list of her species, you can pull out the related taxa or the related species and their corresponding mycorrhizal responsiveness values and get an idea of what might be going on. So pulling a few examples from that data set, if you look at Andropogoniae, so that's the blue stem tribe within the broader grass family Poaceae. So that's things like little blue stem, big blue stem, Indian grass. Those species all had incredibly high responses to AM fungi, that value of greater than 90%, suggesting that these plants were barely hanging on and succumbing after just a few weeks to the lack of AM fungi. And the same can be said about Helianthiae. So that's the sunflower tribe within Asteraceae. That's things like Echinacea that you see here, Silphium and Helianthus, things like that. And so we have some evidence that taxonomically related species are also we would expect little variation within those taxonomic groups. However, this has been rarely tested within genera, and especially in species-rich genera that are occupying different habitats and different niches. I think there's reason for exploration of these diverse genera in terms of their responses to AM fungi. That brings me to this idea of phylogenetic signaling and plant-AM fungal relationships. And just real briefly, phylogenetic signals are the tendency of closely related species to resemble one another to a greater degree in some measurable trait than two species drawn at random from that same phylogenetic tree. And so the phylogenetic signal for plant-AM fungal relationships has been pretty well documented, usually through the context or through the lens of meta-analyses or reviews that are pulling from many different sources, many different papers, things like that. And to my knowledge, this has never been empirically tested within a genera. I think most of that is due to this preconceived notion that species within a genera should be, or within a genus should be very, very similar, if not identical, in their responses to AM fungi based off previous literature. But there's no strong concrete evidence for that. After testing for phylogenetic signaling in a data set or within a group of species, it's also common to test for different models of evolution to see how that trait might have arisen within that phylogeny. So three of the most common models of evolution to test against one another for a trait value would be Brownian motion or random walk evolution. So that trait is evolving kind of anywhere in the phylogeny and any one direction, depending on evolutionary pressures. We also have Ornstein-Uhlenbeck processes, which suggest that there's a constrained trait value that the group of organisms is not easily departing from. And then finally, early burst or rapid divergence evolution. And a great example of that would be Darwin's finches, when that most basal finch species or finch relative arrived on the Galapagos Island to a plethora of empty niches based off food preferences and feeding guilds. We saw rapid divergence in terms of those beak sizes and shapes. So those are 3 common models of evolution you can also test for to determine or get a better idea of how a trait developed within the phylogeny. So that brings me to my study species, or my study system, whichever you'd like to call it, and those are milkweeds, specifically in the genus Asclepias. So the term milkweed, that's kind of a broad catch-all term for any plant in the subfamily Asclepiodoideae within the dogbane family Apocynaceae. So Asclepias and a few other closely related genera are within that subfamily, and so when I say milkweeds, I am just referring to the genus Asclepias. There are about 400 species worldwide, found mostly in North America and northern South America, as well as Africa. Until about 10 years ago, the African species were placed in a different genus, Gomphocarpos, but more recent molecular work has indicated that they should indeed be placed within Asclepias. So that leaves us with about 130 of those 400 species here in North America. And these 8 pictures off to the side, these are all pictures that I took from different parts of the southwestern and western United States that just really showcase the diversity in size and structure of these plants, but all unified under that very unique, very recognizable inflorescence and floral structure. And over the last 20 to 30 years, due at least in part to the concern surrounding monarch butterflies, milkweed ecology has gained a lot of recent attention, but only until about 10 years ago have people began to explore and attempt to understand the relationship between Asclepias and associated soil microbiota. That brings me to the objectives of this study, and that was to first identify the responsiveness of various milkweed species to AM fungi, as well as assessing the root colonization for proxies for abundance of AM fungi in the roots of these species. I then tested for phylogenetic signals of these traits and set out to identify best supporting models of evolution to help predict or gain insight into how these traits arose in the phylogeny. So my initial hypotheses were that milkweeds would display considerable variation in responses to AM fungi, and that strong phylogenetic signals would be detected. So this kind of goes against what the previous literature suggests, but like I said, this has never been tested within a genus, within as many species, or among as many species as I was setting out to test. And just because they're growing in different regions, being subjected or growing in different abiotic conditions, there were just lots of things that led me to believe that there could be more going on than what we might expect. So as with mycorrhizal responsiveness, I hypothesized colonization would also vary, though these phylogenetic signals would probably be weaker just because plants just in general usually don't deviate a ton in their intra-radical AM fungal abundances, especially closely related species. And then lastly, I expected these 2 traits, or hypothesized these 2 traits would likely be driven or supported by different models of evolution. So we'll real briefly cover some methods here. Very straightforward study. Two treatments, mycorrhizal and non-mycorrhizal. Both of these started out with the same sterile background soil collected from Konza Prairie Biological Station, just west of here, about an hour and a half. And the only difference between these 2 treatments are that the mycorrhizal treatment included an inoculum of whole soil. So that would be the entire soil microbial community, and the non-mycorrhizal treatment included a non-mycorrhizal civet. So that same live soil that was used in the mycorrhizal treatment was passed through a 24 micron sieve where the relatively large hyphal fragments and mycorrhizal spores would be retained on top of the sieve, allowing smaller soil microbiota to fall through, and that non-mycorrhizal civet that fell through the sieve added to these non-mycorrhizal treatments. So in theory, the only difference between the pots would be the presence or absence of AM fungi. And I did this for 35 different Asclepias species. And just using the same mycorrhizal responsiveness calculation, there's a couple of different ways to calculate this, but I use the calculation that my PhD advisor used during her time at Konza. And this gives us a value between 0 and 100 if the plant is, if there's any difference in mycorrhizal colonization, or if there's any difference in biomass production, sorry. This calculation often doesn't work for species that are non-responsive or harmed by AM fungi, so things like maybe Brassicaceae or Amaranthaceae, but for species that do benefit, which we had reason to believe milkweeds in general would, this works perfectly fine. Root colonization stained with a trypan blue dye, pretty standard protocol here, and assessed microscopically. These are two pictures of different milkweed species. I think these are both I think this is Asclepias verticillata on the left and Syriaca on the right, looking at AM fungal hyphae that are kind of traversing the length of the root. And so the analyses, again, pretty straightforward, using ANOVAs to test for within species differences between the two mycorrhizal treatments. And then it calculated two different metrics of phylogenetic signal, these being Blomberg's K and Padgel's lambda. And the reason we use these two kind of in tandem is because oftentimes, I should say oftentimes, but occasionally the two will be in disagreement with one another. So when the two do detect strong phylogenetic signals, that gives us better support and stronger support that something is actually being driven by phylogenetic signals. And then tested for models of trait evolution using the package Geiger. So all of this was done in R using PhyTools, Geiger, and a package called APE. So moving on to the exciting stuff, the results. So there is a lot going on this slide. So we'll start over here on the left on this phylogeny or this chronogram, meaning that the branch lengths are relative to evolutionary time. The different colors, you can see the legend down in the bottom. And now that I think about it a little harder, I should probably flip them, but the red indicates low values of responsiveness. So the plants aren't doing that much better in the presence of AM fungi relative to non-AM fungal counterparts. And the blue values indicate species that were highly responsive. And so you can see just looking at this, the groupings, the clusterings of similar colors, which implies that there should be a phylogenetic signal here. What's also interesting about this is this chronogram and color scheme is known as an ancestral state reconstruction. So this actually, you can trace those colors back, or those branch tips back to the nodes and the most recent common ancestors, and that's actually a prediction of what those ancestors, how responsive they would have also been to mycorrhizal fungi. And so we see, starting at the very top of this chronogram, we see a tight clustering of highly responsive species in what we would call the incarnati clade. So those are things like swamp milkweed and whorled milkweed, aquatic milkweed, a few species we can find here in the surrounding remnant prairies and restored prairies. We also see a tight cluster of highly responsive species down towards the bottom with Asclepias tuberosa and Syriaca, so butterfly milkweed and common milkweed, also which you could find around here. So now moving over to phylogenetic signal, we found strong p-values, very low p-values for both of our metrics, indicating that there is a strong phylogenetic signal for mycorrhizal responsiveness within the Asclepias phylogeny that I tested. And then moving down to to the models of evolution, we found the strongest support for Brownian motion through an AIC approach, but those delta AIC values of 2.3 and 2.4 for the other models suggest that those are also relatively competitive as well. So From there, we move on to AM fungal root colonization. So a lot less colorful, lots of greens and yellows, a couple of blues. And so with that, we're predicting that we're probably able to predict or imply that there's not something going on phylogenetically. And when actually calculating those phylogenetic signals, that is indeed true. So not a strong phylogenetic signal at all for AM fungal root colonization. And here's where we see a little bit of that disagreement I was mentioning between the two phylogenetic signal values. So that Blomberg's K of 0.09 is kind of trending towards significance, although not significant, but that really high Padgel's lambda, the disagreement between those two suggests there's something other than phylogenetic relatedness that is likely driving AM fungal colonization. And what's interesting with these models of evolution, we find very strong support for an Ornstein-Uhlenbeck process or that constrained trait value. So mycorrhizal colonization within the root system of the 35 Asclepias species that I tested is constrained to a range of values that these plants likely aren't departing from or deviating from, possibly due to the cost benefits and the trade-offs of the mycorrhizal symbiosis. And so just tying up some loose ends by showing the lack of relationship between colonization and mycorrhizal responsiveness. So just because you have a lot of roots or a lot of AM fungi in your root system doesn't mean you're going to be conferring a lot of benefits, because we know now mycorrhizal taxa vary in their functional traits as well. So in conclusion with this part of my talk, responsiveness to AM fungi is extremely variable within Asclepias and appears to track the phylogeny as an evolved trait through Brownian motion processes, and that forces other than phylogenetic relatedness are driving AM fungal colonization, although we do have strong evidence for selection for an optimum range of colonization values, and little to no relationship between the responsiveness and AM fungal colonization. So that wraps up the first part of my talk, and I'll be finishing up with the influences of biotic and abiotic disturbances on long-term milkweed abundances in the tallgrass prairie. Excuse me. Okay, so grasslands are the most widespread of Earth's terrestrial biomes, and they account for between 30 and 40% of Earth's terrestrial ice-free surfaces. However, this wasn't always the case. Grasses didn't come onto the scene until the late Cretaceous, maybe 66 to 67 million years ago, and it wasn't until about 60 million years later during the Pliocene epoch that we saw these massive expansions of grasslands across the globe. And it's thought that this Pliocene expansion was likely driven by changes in seasonality and rainfall, climate variability, things like that would have promoted fire and other conditions that would select for forbs and grasses over woody species. And we're all very well aware that today grasslands continue to be shaped and maintained by these disturbances such as fire, grazing, and drought. And those are the three disturbances, as well as one other that's kind of facilitated by these three that I'll be talking about. So along with being the most widespread of all of Earth's terrestrial biomes, grasslands are also widely considered to be among, if not the most threatened, and that's very evident here in the tallgrass prairie in central North America, not only because of agricultural intensification and urban sprawl and things like that, but the remnant prairies, the intact grasslands that remain today face these looming threats that are likely driven or mostly driven by alterations to those disturbance regimes that I mentioned in the previous slide. So reduced fire frequency, that's increasing woody cover on the landscape. Removal of large ungulate herbivores has increased C4 grass dominance. So you think back to the Pleistocene, maybe 12,000 years ago, there was much more ungulate herbivore biomass on the landscape that was selecting for C4 grasses. And so without those, we're seeing a greater dominance of these C4 grasses than what would have been historically. And then we're seeing increasing climatic variability that's causing species reorderings in some cases with selection for those more drought tolerant and more drought resistant species. And so I've already talked about milkweeds and Asclepias pretty extensively in the previous part of my talk. So I just want to jump straight into my study species for this part of my talk. And those are Asclepias viridis, Asclepias syriaca, verticillata, and tuberosa. And one thing that I want to point out is where these species are typically found. So Asclepias viridis and verticillata, so green antelopehorn and whorled milkweed, are found almost exclusively in the southern and central Great Plains. That's definitely the heart of their range, whereas these other two, common milkweed and butterfly milkweed, can be found in the Great Plains, but also east to the Atlantic coast. Excuse me. And that will come into play here in a little bit. So the objectives of this study were to assess the effects of the following disturbances on the long-term cover of these 4 species. And those disturbances are fire frequency, bison grazing, I'm throwing in woody cover as well, and drought. So I hypothesized that the greatest cover of milkweeds would be found at intermediate fire frequencies, while grazing would promote greater milkweed cover due to competitive release from grasses, with the herbivores selecting for those C4 grasses. I hypothesized milkweed cover would be negatively associated with woody cover, and that the 2 species that are found almost exclusively in the Great Plains, I assumed they would be relatively unaffected by drought, while these other 2 species, although they do have broader ranges, we are kind of at the western extent, and you expect more severe effects of climatic alterations at species range extremes. So that's why I thought potentially these 2 species would be more negatively affected by drought. This data was compiled from Konza Prairie Biological Station, the NSF LTER site that most of you are probably familiar with, just west of here. Konza is divided into 57 watersheds based off topographies with varying fire frequencies and grazing treatments, and an average watershed area of about half a hectare or an acre and a half. And so I just want to detail some of the plant community sampling protocols on Konza Prairie. So these watersheds that I mentioned previously are divided into upland and lowland sites, so based off topography. And in each of these topographic positions, there are 4 50-meter transects with 5 permanent plots along each transect. And typically the plant community sampling is conducted in May and August of each year since 1983, and that captures the phenology of the plant community pretty well. And so within each of these permanent plots, Daubenmeier cover classes were recorded for all species present. So it's a presence only metric. For the sake of my study, I trimmed those years down to 1991 to 2020 to better align the bison grazing and fire frequency in watersheds of interest. So I'm conversing with some Konza researchers, Zach Radizak mostly. He suggested that 1991 would kind of be the best year where the bison numbers start to level off and you start to really see the effects of the grazing. I took midpoint of the cover classes for each species and averaged across plots with transects as my replicates. So that's for the milkweeds. And then for woody cover, I did the same, but summed the cover of the 6 most common woody encroachers and averaging that across transects as well. For my analyses, I used GAMs, or generalized additive models, in R, and these models are great for fitting these complex time series data sets where you might have multiple error distributions within the same data set or data frame. And I used simple univariate models to tease out the individual effects of fire, grazing, woody cover, or drought, with time as my smoothing term for these GAM models. So moving on to the results, we'll start with Asclepias viridis or green antelope horn, because that was through my analyses the most abundant species on Konza Prairie. So each of the following slides will be similarly structured to this, with each slide being species specific. So that's a close-up, really nice picture of Asclepias viridis. I think it often gets overlooked, but those kind of cream white corollas and purple nectaries really stand out if you take a second to look at them. So this first figure is always going to be fire frequency. So along the x-axis, we have year to 2020, and then along the y-axis we have peak cover. So Asclepias viridis is only making up between 0.5% and 1% of the total plant community cover on the landscape. But as we can see here, a steady kind of consistent abundance in the 1-year fire frequencies, where we saw pretty strong decreases in the 20-year fire frequency. So that's fire happening once every 20 years. And then pretty steady also in the 4-year fire frequency, although the abundances were initially lower relative to the 1-year fire frequency. So next, we're going to look at grazing. So the blue bar indicates the presence of bison, and the red line and ribbon indicate the absence of bison. And so they started out in these watersheds with there being greater abundances in the non-grazed watersheds, but that dropped off pretty dramatically through time, where we saw the opposite pattern in the grazed watershed. So there is some evidence of maybe some competitive release from grasses here for green antelope horn. And then this pattern couldn't be any more clear. With increasing woody cover, we saw dramatic decreases in Asclepias viridis abundances on the landscape. And so that's what I have for Asclepias viridis. Now we're moving on to Syriaca, or common milkweed, the big common clonal species that you might see in roadsides here. So that's a close-up of its inflorescence, usually not this deep purple or magenta color, usually more of a kind of cream pink, but also a very, very pretty plant. With fire frequency, we're seeing decreases kind of regardless of fire frequency, although those decreases are more apparent and stronger in that 20-year fire frequency, and really no difference by the end of the study in each of the 3 fire frequency regimes. And then a similar pattern with grazing, regardless of whether bison were present or absent on the landscape, we see decreases in common milkweed abundances through time. And this was really interesting to me, and I still haven't quite put my finger on it, but there was actually a significant increase in Asclepias syriaca abundance as woody cover increased as well. And I don't have too many leads on this, so I'm open to suggestions on what other people might think for that interesting result. Unfortunately, I don't have a close-up nice picture of Asclepias verticillata, but we're seeing similar patterns. Here's what we saw with Asclepias viridis, so the green antelope horn. So although it started out at a higher initial abundance, we see this greater abundance maintained in the 1-year fire frequency, but pretty consistent, albeit lower levels in the 4 and 20-year fire frequencies as well. And then we're kind of seeing a flip of patterns here, where we had initial greater abundances in the bison grazed watersheds, but that's decreasing through time. And there is some evidence that at least for cattle that they will readily consume Asclepias verticillata. I don't know if that's the same for bison, but that's one theory that I have here. And in the absence of grazing, there is a slight increase. So it'd be interesting to see how this plays out through time. And then finally, we see, again, similar patterns to what we observed with Asclepias viridis, where we see a significant decrease, much wider confidence intervals here, but this was a significant decrease in Asclepias verticillata abundance as woody cover increased through time, or as woody cover increased. So time is not factored in here. And then finally, last but certainly not least, Asclepias tuberosa, probably my favorite just based on the color. But definitely the least exciting in terms of results. No effective fire frequency. This could be due to the low abundances. If you look at the y-axis, only maybe 0.3% in terms of cover. And then no effective grazing. And this very similar to Asclepias striaca, but this effect was not significant here with those confidence intervals overlapping 0. So a lot of people came up to me after my dissertation asking how woody cover was influenced by fire frequency, and it's sad, but I'm glad the results turned out the way they did. In a 20-year fire frequency, you can't argue with that pattern, with woody cover increasing dramatically through time, and even increasing with the 4-year fire frequency as well, which has been suggested by Konza researchers in recent years, that 4 years might not be enough to maintain or reduce reduce woody cover. And then essentially a flat line with the 1-year fire frequencies there. So now moving on to drought. I've been talking a little bit about drought for this project, and this will be species by species. So first here I have Asclepias viridis, and along the x-axis we have SPEI, that's standardized precipitation evapotranspiration index, and you can interpret this x-axis as standard deviations from norm. So 0 being normal precipitation year. Anything less than -1 would be considered extreme drought. Anything greater than 1 would be an exceptional wet year. And so I did this with 3 different windows and grains. And so panel A, that 3-month interval, that indicates the 3 months leading up to the growing season. So February, March, and April leading up to the May growing season. 12 months would be the previous 12 months, and then 36 months would be the previous 36 months to capture, to see if these plants were maybe more sensitive to long-term drought compared to short-term drought, because they are these long-lived perennial species. And although we think we might see a pattern here with increasing abundance as years get wetter, these were actually not significant either, with confidence intervals pretty strongly overlapping 0. And then moving on to Asclepias syriaca, again, really no effective drought. We think we might see some patterns, but nothing significant. And as well for Asclepias verticillata and tuberosa. So in conclusion, really no effective drought through this analysis on milkweed abundances through time. So in conclusion, Asclepias viridis and verticillata appear to be incredibly sensitive to these altered disturbance regimes, which could likely be linked to increased woody cover on the landscape. The other 2 species were relatively unaffected, with the only significance being the fact that syriaca significantly increased with woody cover. And then I do want to point out that Asclepias syriaca did appear to be decreasing in abundance regardless of disturbance regime on the landscape, which I think has a lot of potential implications for monarch conservation in the Midwest. And so with that, I'd like to acknowledge the funding agencies for this research, and the faculty members and grad students that helped me along the way. And I just want to end with what I'll be doing at the Kansas Biological Survey, and Peggy kind of highlighted these. Currently working on the MAPS project, looking at plant cohorts and microbial communities on ecosystem services, and then that pending USDA grant that would allow me to look at the soil microbial and plant diversity and their influences on ecosystem services. So with that, I'll take any questions.
Thank you, Eric. That was a really nice presentation.
Thank you.
I have several questions. I'm sure lots of there are other questions in the
I
audience.
saw you doing a lot of writing.
First, a big round of applause. So now I need to distinguish raised hands from clapping hands. All right. Well, I'm happy to start with a question, and that is, did you look at your data from like a landscape perspective with all the species? Like, it seems as though heterogeneity in management in tallgrass prairie might actually sustain, all of the species. What are your thoughts on that?
Oh, no, I didn't assess them like that. I simply used those what I was calling disturbances as categorical variables. So that would be an interesting way to look at that. And I'm just trying to picture how to incorporate that into an analytical framework.
Mm-hmm.
Um,
Yeah, I was just thinking, looking at all the transects, averaging over time, as long as they're equitable.
and I think that was the limit with this, was lining up, because I knew I wanted bison and I knew I wanted fire frequency, and so I had to selectively remove a bunch of the watersheds that didn't fit those a priori criteria,
Mm-hmm.
because they didn't match up well.
Helen?
Thanks. Yeah, so Liz and Jim and Peggy's lab know far more about this than I do, but I know that they've shown that more mycorrhizal responsiveness with late successional species. And I've worked with Asclepias medii, so I noticed that was, kind of in the middle there. Did you see any general ecological patterns?
So I am doing some follow-up analyses. That's why this study hasn't been submitted yet, but no. So this actually all came about because I started this study with about 10 species, and I thought, these are much more different than what I would have expected. But that kind of facilitated and prompted other questions. So I looked at different, trying to find different ecological patterns, whether they were clonal, whether they, what area are they native to, things like that, soil nutrient requirements. And the thing I landed on was lining it up with a phylogeny, and sure enough, like they broke out very cleanly in terms of where they fell on the phylogeny and how responsive or dependent they were. And I haven't done anything with, as far as like, I guess you could look at coefficients of conservation, but that's not something that I've done or looked at.
Chip Taylor and Eric, if you don't know yet, since you're new, Chip Taylor is the director of Monarch Watch.
Hey, Eric.
Yeah, I worked with Kristen Baum for my PhD as well.
Yeah, well, I need to call Kristen and give her a talk. Various things. Yeah, she's doing great work down there in Oklahoma. We appreciate what she's been doing, and she has some nice plots where she is trying to maintain milkweeds, which is a really a good thing, especially in Oklahoma where they try to mow everything down along the roadsides. So there were some really interesting things that you had to say. One of which was that the woody relationship with Siraca. I don't know how to explain that either, because it seems to be counterintuitive relative to what we know about Siraca, which is that it's a disturbance-related species. It's a pioneer species that likes to follow disturbance, and it is not a good competitor. So your result just seems to go against everything that we think we know about that species.
And so the only thing that I've been able to come up with, and I mean, I guess you take it with a grain of salt, but I did find I think it was a Xerces Society flyer or report or something suggesting that Asclepias syriaca is the most shade tolerant of the native grassland or prairie milkweed species. And anecdotally, I have seen it on forest edges in Oklahoma, but I don't have any empirical data to back that up, other than what I just showed you.
All All right, good. Well, I have another observation and that kind of fits with your bison grazing, because if you really want to see viridis in great abundance, you go to a horse pasture or a cow pasture, because they do not graze on it, but they will graze on virtually every other milkweed in those pastures. So there is a strong herbivore relationship between what you see in pastures and who is doing the grazing.
Yeah, definitely.
Yeah. Some of the densities of viridis in horse pastures and cow pastures are simply amazing. I mean, we're talking about thousands of plants per acre.
Yeah.
Just astonishing.
Peggy?
So I just had a question, Eric, about, when you were doing the work in the second part of your talk, it looked like, some of the milkweed species did best with the 1-year fire regime. And I'm wondering, how likely is it that any area was burned every year? Because it seems like that would be pretty unlikely.
Yes. So, and I think the issue lies within the 1 year versus the 4 years. So talking with Zach and some other researchers from Konza, the 4-year fire frequency, as I suggested or hinted at, isn't actually keeping woody cover at bay. They're still experiencing expansion. And those annual fire frequencies, I mean, it's not really reasonable, but I know a lot of landowners still do utilize that. And it seems to be a pretty selective practice too, because we often see reductions in broadleaves and in diversity when using those annual fire frequencies. But I would bet that there would be greater abundances in maybe a 2-year, every other year. And that would be, I think, much more sustainable.
And would that be reasonable for historically? I mean, I'm really thinking of historical patterns here, and historically things have changed quite a bit, right? So then historically,
yeah, and that, I guess that kind of goes back to what I was saying. I think by most accounts, between 4 and 10 years in the tallgrass prairie, 10 would really be pushing it. But that was also in the absence of all these other anthropogenic factors and forces.
Yeah. Thank you, Eric.
Jude?
Thanks, Eric.
Yeah.
Could you go back to the, I'm going to go on the Syriaca as well, to the first ribbon plot.
There?
I don't know. Yeah, you need to share your screen maybe.
Oh, oops. Let's see. I can do that. There we go.
Yeah. I guess my question had to do is like, what's the explanation for the big disparity at the start in 1990 from the 20-year plots to the other two? I mean, why were the populations so different among those sites?
So I could take this data back to 1983. That's when it started, but it didn't start in 1983 for all the plots that I was interested in, or all the watersheds of interest. So I have given that a lot of thought in scaling that back. Otherwise, I think it would just have to do with inherent or innate patchiness on the landscape. I really don't, because we see that pop up a few times with the initial abundances in 1991 being higher in one treatment and actually kind of staying flat. So we can't really indicate that treatment's maintaining higher abundances if it's flat for 2 or 3 treatments across time. But yeah, I think if I took it back to 1983 and there were differences there as well, I think it would have have to do with just the patchiness on the landscape.
All right, I'll take that. Thanks.
It is patchy out there. Rondi?
You're muted.
Yes. You can hear me?
Yeah.
Oh, thank you. Great talk. I was
Thanks.
actually interested in in one of your last data slides where you're referring to the responsive drought. And it seemed like drought didn't really affect any of the species used in the study. And I was interested because verticillata, which was a species that was in that study, is a very drought tolerant species. It grows in xeric soils and it has a very fibrous root system, so very fine-like roots which allows it to remove residual moisture from basically tight, poor pockets. So it's very good with low moisture in the soils. So I was wondering, is it a possibility that you saw similar responses? I'm trying to figure out why they might have been similar across species. Could it be that all the species were being observed in sympatry, so therefore there weren't like these different habitats at play? Like, for example, in these xeric soils that's very dry, would all of the species behave the same? Whereas if you had them in a sympatry, which is more of like a common garden where everything is kind of plentiful, but you just cut back the moisture, the soil or endaphic environment may not have resembled what you might have been observed in nature somewhere out there.
Yeah, that's a very interesting point. So they are, yes, they're certainly growing in sympatry, but in each of these plots rarely are multiple species within one of these relatively small plots. And I haven't looked at or thought of physiological or anatomical structures that would maybe inform these results. I use those different time links, those different links, because I simply wasn't sure if short-term or long-term drought would be more impactful on a particular species. And the thing I keep coming back to is that it didn't seem to matter at these scales. And my initial thought is that they're long-lived perennial species that are well adapted to growing in dynamic grassland environments that have historically experienced periods of drought. That's not to say it's not going to be worse with more persistent or more frequent drought in the future, which could be informed by greenhouse or growth chamber studies, but I don't know.
Thank you.
Chip's got his hand up again.
All right, I'm unmuted now. All right, one of the things that we have seen and we hear a lot of complaints about with regard to Syraca is that it's very susceptible as a clonal species to fungal attack, and where huge clones are just taken out of the system within a year. I mean, and I've seen it myself where I've been familiar with large clones that have they're just gone from one year to the next. I mean, these are clones that are probably in some cases 20 or 30 years old, and they simply disappear overnight. And I wonder if you've been aware of this or factored any of this into your thinking about the changes over time.
No, I actually haven't given that any thought until you just mentioned it. I know at Konza, there's, I guess now thinking back when I have been out there, there have been patches that have shown more necrosis or yellowing. I didn't know that could be a fungal pathogen or might be a fungal pathogen.
It is.
Okay.
And it's a pretty serious one, and it seems to only affect Syraca among the clonal species. Sullivantii doesn't seem to be impacted on it. I've never heard of anybody talking about Sullivantii responding to this yellow leaf sort of thing. I'm trying to remember the name of this thing, but it's a pretty common fungus. Anyway, yeah, so this is another aspect with regard to Syriaca, but perhaps not the other species.
Gotcha. No, that would be very interesting to look into.
All right, Eric. Thank you very much for presenting your work. And wait, Chip, you don't have another question, or is your hand still up there?
Well, my hand was still up there, but Eric, I'll send you a picture of Verticillata, okay?
That was good. Thank you so much.
Okay, I think Ashley has one too. So I have two comments. One is a request. Eric, it would be great if you could teach me in my lab, and there are probably other people here in the center that are interested in learning about the generalized additive models. So I'll be talking to you about that. And then I want to remind everyone that next week we're having another research seminar. It will also be remote. We're going to continue with the below ground theme, but we are going to switch to invertebrates, and Matt Callahan from the USDA Forest Service is going to talk about earthworms and fire. So that should be a pretty fun talk. So anyway, thanks everybody for coming. I hope you have a wonderful weekend.